really to emphasize very heavily how important genetic findings have become in contributing to the evolution of this classification, and how really, in practice today, we actually cannot make these diagnoses reliably without molecular genetics. And then I want to actually go through some of the methodology that can be used for molecular genetics, because there are many different assays that one can use. And so I'll try to highlight the pros and cons of these towards the latter half of the talk. So we all know that childhood tumours are different from adult ones. 2/3 of childhood tumours include brain tumours, leukemias and lymphomas, and of the remainder, about 1/3 are undifferentiated. And generally speaking, undifferentiated cells tend to be quite blue. But many of the other cells in the other tumours, particularly leukemias and lymphomas, of course, are also blue. So you could argue that there are way more blue cell tumours than just undifferentiated sarcomas, but I have to restrict myself to a group for today, so that's the group I will talk about. So this image will just, or this slide will just show you a number of images of blue cell tumours of multiple different types. So on the top left we have what is actually a Burkitt lymphoma invading the smooth muscle of the bowel. And you can see mucosal necrosis overlying it. That's made up of blue cell tumours. Just below that on the left we have blue cell tumours that are actually blastema. And if we look around, in this case we can see tubule formation. So that's obviously very helpful. But sometimes you get just blastema. So on the top right we have a Ewing sarcoma. So a very monotonous blue cell population. And on the bottom right, we actually have an undifferentiated neuroblastoma. So really all of these tumours are made up of blue cells. Just in the middle at the bottom, we have another case. This is from a nephrectomy. You can see the fat, the capsule, some compressed renal parenchyma, tumour capsule, and then deep to that, blastema, a particular Booth pattern. But if you have a needle core biopsy of that, you may have simply just blue cells as well. So for the purposes of the talk, I'm going to restrict myself to undifferentiated small round cell sarcomas of bone and soft tissue. And really these come in 4 major categories. We have Ewing sarcoma, or if you like, classic Ewing sarcoma. And then we have a number of other entities which have been historically described as Ewing-like. But as we understand more and more about the molecular and the behavior of these tumours, we have categorized these into a total of 3 additional categories. So we have the round cell sarcomas with EWSR1 non-ETS fusions, so not FLI1, ERG, or the other members of that gene family. We have the sick rearranged sarcomas, and we have the sarcomas with B-core genetic aberrations. So the classic Ewing sarcoma is a remarkably monotonous blue cell tumour. So when we get, for example, a bone biopsy, we will roll the bone core on a slide, or if soft tissue is a bit easier to get cells to stick, because sometimes in a bony tumour they're hidden behind the matrix if it's a small cell osteosarcoma. But if you find that you're getting a very cellular population of round blue cells and they're remarkably monotonous, Ewing has to be very high on your list of differentials. You don't get that monotony in alveolar rhabdomyosarcoma sarcoma, and you don't get it in osteosarcoma at all. And even in the variant forms of small round cell sarcoma, as I will mention in a moment, you tend to see a little more variability in the nuclei. So on the top left, you can see a touch imprint that I made from a core biopsy. We get our samples fresh, and I'll come back to that later on. There's a huge value in getting your samples fresh. You can do a lot more with the tissue, so you can immediately get an idea of what you're dealing with. The fixed tissue, of course, you still need histology, you still need immunohistochemistry, and you should never bypass these and go straight to molecular, because that may lead you down a false path. The fixed tissue sections are shown in low power in the middle part of the left-hand side of the slide, and at higher power in the middle. And we can see that at the bottom we have a lot of very thick fibrovascular cores and blue cells associated with this. This pattern, this architectural pattern, we see really only in Ewing sarcoma or alveolar rhabdo. But when the cells are so monotonous, Ewing has to be top of your differential. These tumor cells will stain with CD99 in a very crisp membrane-associated pattern that looks like a chicken wire or a mosaic. So we get CD99 staining lots of tumors, but if we have this very crisp staining, that is characteristic of Ewing sarcoma. And on the bottom right, we have nuclear staining for NKX 2.2, which is very typically positive in classic Ewing sarcoma. So there are a number of translocations that are possible. You can see that the one partner is typically EWSR1. It may be fused with any one of a number of genes from the so-called ETS gene family, which includes FLI1, ERG, ETV1. The vast majority are EWSR1, FLI1 fusions. They make up about 85% of Ewing sarcoma. In certain instances, we get a FUS gene rearranged with these ETS genes instead of FLI1. FUS belongs to the same family, so biologically this makes perfect sense. Now, with Ewing sarcoma and also with osteosarcoma, one of the important histological features is to look for the percentage of necrosis post-chemotherapy. And here I'm showing you a distal femoral resection from a patient post-chemotherapy, and the patient had a diagnosis of Ewing sarcoma. So we get the sample, we take photographs of it, we ink it, we remove any soft tissue that might be attached to it, and then we take a bone saw and we cut right through the bone. We look at the imaging first and work out where is the best plane to cut in. And we take one full face, you could say a carpaccio of the bone. I mean, you may want to slice it in many slices, as I will show you in a moment. And then you can lay it out like this and have a look at it. This one is unusual because it's got this white material, that's a cement-like material that was put in to prevent fracture following presentation. And the surrounding tissue is somewhat white, somewhat yellow, suggestive of probably regressive changes. And then we have to sample one entire face in order to evaluate the percentage of necrosis. You can see that we've made a grid diagram, and this starts at A5 simply because we would have had some soft tissue to sample in A1, 2, 3, and 4. But the point is that we are looking at one full face and we're evaluating the percentage of necrosis in the tumor, which is very important prognostically. So the original biopsy was the one that I showed you on the very first slide. It's a Ewing sarcoma, completely viable. Post-chemotherapy, in this particular case, we have 100% necrosis, which is a good prognostic feature. But that's not the case for all round cell sarcomas, and particularly when we get to the variant translocations involving EWSR1 and some other non-ETS fusion, we see that there is no response, no histological response to neoadjuvant chemotherapy in the group where we have EWSR1 rearrangement with NFAT-C2. And you're not going to know this just from the histology. You may suspect it if the cell nuclei are a little bit more variable than in your typically very monotonous Ewing sarcoma, but you need to prove it molecularly. So again, these cases will tend to stain with CD99, but it's not as consistently so, and sometimes they stain with CD138 as well. The other group in this ETS or EWSR1 non-ETS fused small round blue cell sarcoma is the PATS rearranged tumors, and these may be much more polyphenotypic in terms of their immunoreactivity, and in this particular scenario the chemotherapy response tends to be poor as well. So it's good to know that up front and potentially just go for resection, which may be curative in the case of NFAT-C2, but you may also get late metastasis, but you should not in these cases expect a great chemotherapy response at all. In fact, you may get none. Does this actually mean anything, finding these different translocations? Well, if you look at a deeper level at these tumors, either by methylome or by transcriptome, to see what is expressed in these tumors, there are differences, and we can see that on heat maps. So we get clustering of the tumors according to the translocation associated with them. And we can also see it in a principal component analysis plot on the right-hand side. You can see that there's clustering of the B-core tumors, the NFAT-C2 tumors, the Ewing sarcomas, and the sick rearranged sarcomas separately from one another. And this is borne out also when we look at the transcriptome of these cells. They do not all cluster together. So they have a different transcriptional profile and different biological behavior as a result, including different chemo response. The third category is the sick rearranged category, and this tends to occur in young adults, but also in children, and we have had some cases like this. It's more common in the soft tissue by far, but it can actually occur anywhere, including visceral sites. And again, I would say Don't think that anything cannot happen in a particular location. We've had a desmoplastic small round cell tumour of salivary gland, we've had a primary one in bone, we've had a primary synovial sarcoma of liver. So the most bizarre anatomic locations are still possible for these tumours. These tumours tend to show diffuse growth of spindles, so they're not always completely round, or epithelioid cells with some lobulation. So again, these nuclei are not as monotonous as you see in the classic Ewing sarcoma. And there may be a myxoid stroma in the background. CD99 is usually patchy, so it may be very patchy, only small clusters of cell staining as opposed to the chicken wire that you get with the classic Ewing sarcoma. And importantly, WT1, the Wilms tumor 1 gene protein product, is positive in these cases, so that we do not see in classic Ewing sarcoma. So that's also a clue. So if you're seeing a case that doesn't look like your classic Ewing sarcoma, you do your basic immunos, that'll already guide you towards what you might be dealing with here. Most of these cases have a sick DUX4 fusion, but rarely we have alternative partner genes involved with sick as well. A caveat that looking for the sick rearrangement, and I'll come back to this when I discuss the methodology, is that not all techniques will detect this, so it may be cryptic by fluorescence in situ hybridization studies, and it is known to be difficult to pick up by NGS as well. So in the end of the day, you may end up doing a PCR assay instead, just to be sure. Here's a case that we had in the last number of years. It was a young boy in his early teens who had a large mass, as you can see, the lobulated white mass there. He underwent chemotherapy, had a resection, so the middle image is not the post-chemo image. He actually, I'll show you the histology in a moment, he did not respond very well, but he had a fairly extensive plastic surgery to take this out, so plastic surgery and oncologic surgery were involved in this case. But unfortunately, he relapsed on the contralateral side, just not far from the midline. And this is what the histology looked like. So a round cell sarcoma, a bit more variable in the nuclei than you would see, and some of them you can see are lobulated, some of them have nucleoli. So these are already clues that this is not a classic Ewing sarcoma. CD99 shows a very strange pattern, a lot of cytoplasmic staining. It's not just that crisp chicken wire pattern that I showed you earlier, and not all nuclei are staining, or not all cells are staining. WT1 is very strongly positive, so again, this is a clue to us that we may be dealing with a sick rearranged sarcoma. And unfortunately, post chemotherapy, he really has a very limited response, and that's typical. These cases do not do well. They tend to relapse, and they tend to have very poor outcomes generally. And then the fourth category, again small round blue cell, is the B-core rearranged group. B-core rearranged sarcomas may have a translocation where B-core is rearranged typically with CCNB3, or we may have an internal tandem duplication of the B-core gene. This actually underpins a whole host of different tumours, not all of which are small, round and blue. So it includes clear cell sarcoma of the kidney. Actually, that's also in the vast majority of cases driven by a B-core aberration, and also undifferentiated round cell sarcoma of infancy. Far more common in males, as is the case with CCSK as well, of course, and they're somewhat more common in bone, especially long bones and pelvis. The characteristic immunostaining, and we had a little discussion about this yesterday in relation to one of the cases that was presented. The characteristic trio is B-core TLE1 and cyclin D1 immunostaining, and SAP-B2 may also be highly expressed, but that can also be expressed in osteosarcoma. In the B-core CCNB3 rearranged cases, we tend to get very high expression of CCNB3, which can be picked up by immunohistochemistry as well. B-core is a subunit of a polycomb repressor complex, which is involved in histone modification and therefore has an epigenetic effect. That's just an image of the complex with B-core indicated in green, and the effect it has on the chromatin, where the histones are marked with a silencing mark and genes are turned off, if you like, as a result. And then the various tumors that may be underpinned by this. It's not just small round blue cell tumors. It's actually a particular interest of mine. My research group looks at B-core as our major focus. Here's an example of a core biopsy from a case. It shows round cell sarcoma. not particularly blue, you might say. It seems to have quite a lot of cytoplasm. But the B core immunostain is very strongly positive, and when it's strongly diffusely positive like this, we should be thinking about a B core associated tumour. B core can stain a lot of things, but if it's strong and diffuse, think B core tumour. So immunos do take us a certain part of the way, the combination of morphology and immunos. But really, as I said at the beginning, in today's practice, without molecular, we're not really working at the gold standard of diagnostics. This particular case is a desmoplastic small round cell tumour. You can see there are nests of blue cells, and immunos here are very helpful because we get the rearrangement of EWSR1 with WT1, and therefore, because of the rearrangement, we lose the N terminus of WT1, and that's negative, but we still have retention of the C terminal antibody expression. So again, this will certainly suggest the diagnosis of DSRCT in conjunction with probably the clinical picture in the patient and the histology. I'm showing you a very classic case, but also the immunos are not always reliable because you can get cryptic rearrangements where the immunos don't show the expected pattern. So in the end of the day, what you really need to make a firm diagnosis at gold standard level is molecular. So what is the best way to handle your specimen in order to come to the correct diagnosis? And this is sort of the second part of my talk. Firstly, in our department, since I started there, we have been getting all of our biopsies fresh. And the reason for this is that if you get a biopsy already in formalin, you actually can't do anything with it on the day in question. You can only stick it on the processor, it'll come out the next day, and you don't know at this point, are you dealing with a lymphoma, in which case you might want to do flow cytometry, or are you dealing with a particular blue cell sarcoma, or what are you dealing with? So just knowing that much from a touch prep, like I showed you with the Ewing sarcoma, staining up with H&E, and having a look at it informs you quite a lot as to what you're likely to be dealing with. It's not your final diagnosis, but it can certainly give you some ideas of what you want to do with the case going forward. You'll then fix it and process it for histology and immunohistochemistry. But before you do that, you can save a little piece, and often these tumours are massive, you only get a small core of material. But nonetheless, you can snap freeze a small bit of that core in a cryovial, plunge it into liquid nitrogen, into your -80 freezer, and you have protected the nucleic acids in that tissue to the highest level that you can. So if you then want to extract these nucleic acids and run next generation sequencing, for example, you have the material available. So when you get your histology the next day and your immunostains, you will then be able to go back to that material and use it, and you have the best quality material to interrogate. Yes, things can be done out of paraffin, but we're more limited in what we can do. Interpretation is more difficult, and extraction is actually more laborious as well. Flow cytometry you can't do anymore when you've fixed your tissue, so if that's even a consideration, you should know it from your touch preps and be able to give your flow lab some material, and they may get you an answer that very day. But you still have to do your histology and immunohistochemistry every time. That should never be bypassed. So what is it that we might want to look for, and how best should we look for it? Well, chromosomal translocations we can look for by FISH. For instance, in situ hybridization, you need to have the probes that will interrogate specifically the translocation that you're looking at, and these can flank a breakpoint and therefore show a split signal if you have a rearrangement, or they can be on either of the partner genes that come together and give you a fused signal if you have a rearrangement. RT-PCR, real-time PCR, is a very quick method as well, and particularly if you're extracting from fresh material, you'll get good quality nucleic acids, and it's a quick extraction. Of course, you can also look for chromosomal translocations by next generation sequencing. Gains and losses, so this would be more important in, for example, nephroblastoma and neuroblastoma. This can be done by FISH, and again, if you have your fresh material, you can simply dab your core of material onto glass slides. Entire cells will stick to the glass slide. You can air dry it and give it to your cytogenetics lab if they can do FISH for you. If we do that and get it to the lab by 3 p.m, we will have an answer by lunchtime the next day. But you need to know what you're looking for, because they will not look with every probe for every potential rearrangement. And sometimes you may think it's going to be something, and you may find you have to do a second round. So be prepared for that as well. But certainly that's a technique that I would highly recommend, because it's quick and it's not that difficult, especially on fresh tissue. If you do it on paraffin, you're cutting through nuclei and you have to count way more nuclei to get a reliable answer as to whether you've got a rearrangement or not. Point mutations, such as driver mutations in a cancer or small insertions and deletions, really you need PCR-based techniques. With sequencing, ideally, next generation sequencing will do it for you as well. Now, in terms of having multiple assays on one platform, we have moved to next generation sequencing. Actually, it was fortuitous during COVID, because of social distancing. Actually, we were allowed to buy a porta-cabin so that the scientists in the lab had more space and didn't have to be crowded on each other, and that was an opportunity to have more space and to be able to then invest in next generation sequencing. So that is something that we've had in the last number of years. So in terms of translocations, this is a very typical list of the translocations that tend to segregate with particular tumor types, and the gene event is shown on the right. This is all very well known, and a diagnostic table, if you like. With fish, it works best on fresh frozen material, and not so great on fixed material. You don't need to have viable material though. So historically, we used to culture the cells, do a metaphase spread, and then look at specific chromosomes. That is not essential. You can look essentially blindly at these with just the probes. It doesn't matter where they're sitting as such. One probe can cover several assays, and again, if we just go back to this, you can see that in many instances EWSR1 is rearranged, and so if you have a good idea of what tumor you're looking at, you can confirm a rearrangement of EWSR1. It doesn't tell you which partner gene it's paired with, which you may want to then follow up more specifically. The cons are that you will find only what you're looking for. That's true of all of these individual assays. So if you're looking for something that isn't there, you're going to get a negative result. You may want to go back and try another assay, and you may end up losing time doing it that way. So this is the real beauty of next generation sequencing and having a panel of genes to look at. Just to go through the technique of FISH, so basically, as I say, you've dabbed your tissue onto glass slides, cells have stuck, you've got entire nuclei sitting on the glass slide. This is the target cell, the DNA, you won't see it, but it's depicted as double-stranded black bands right there. And so in the cytogenetics lab, these slides will be heat denatured to open up the DNA, and then a probe that is fluorescently labeled will be spread on the slide, and the excess probe will be washed off. The probe should anneal very specifically just to the DNA sequence. This is DNA that you're looking for, and then you wash off the excess. You look at it under a fluorescent scope, and you see, in a normal situation, 2 copies of any gene, so that's wild type. As I say, historically this was done on metaphase spreads, so fresh tumor material was cultured, the cells were arrested in metaphase, and here you can see a 12-15 translocation, so with chromosome 12, chromosome 15, and here's a fusion where we have a rearrangement. This is a case of congenital fibrosarcoma. This is what the interphase fish will look like. So here we don't need to culture the cells, we simply dab the tumor cells onto the glass slide. And again, we're looking at a particular rearrangement. In this case, it's SS18, SSX rearrangement. We have a break apart probe, so the probe in the normal context should flank the break point where the break usually occurs, and we get a fused signal of green and red, which is yellow. When we have a rearrangement, the other chromosomes are rearranged, and we get a separation, a break apart of these probes. So we know that there's a rearrangement, the chromosome is split at that side. This is an example of collagen 1 alpha 1 PTGF beta. So this is a 1722 translocation. I mentioned this yesterday in the context of DFSP. And this is a case where we can see amplification of a fusion. So this is a PAX7 fusion, and we get amplification of the signal. So you can see lots and lots of green dots. There should really only be 2 in a normal cell, 2 copies of the gene, but here we're getting amplification, so a lot of green dots. Of course, we use this for other tumours as well, not just small round blue cell sarcomas. And in the case of neuroblastoma, we will do this every single time, look for MIC-N rearrangement, MIC-N amplification. And this may come in the form of double minutes, where we have lots of copies of the NMIC gene scattered all over the nuclei, or homogeneously staining regions where we get a string of the same copy of the gene, multiple copies of the gene, at the correct address, if you like, on the short arm of chromosome 2. So this is a case where we have double minutes, very high level amplification. The nuclei are counterstained blue. We have a fluorescent red MIC-N probe, and you can see there are multiple copies here. So this is not normal, this is very high level amplification. RT-PCR can work from fresh frozen tissue or from paraffin. We don't require a lot of material for this. It's extremely sensitive, so you need to have your lab be very careful not to get false positives. You may get false negatives if your primers are not designed to encompass variants in where the breakpoint may occur. If you only go for the actual most common, and we do see variants. But you can go back to the material and try it again, so that is very useful. And this is what RT-PCR may look like. So that's for synovial sarcoma. So we have a housekeeping gene that we test for to show that there is adequate quality of nucleic acids within the material. This is done on RNA, which is then reverse transcribed into cDNA, and then we get a real-time amplification. So we do still use real-time PCR in our lab. That was the mainstay of what we used before we had NGS, because it's very quick, and if we're waiting to accumulate enough cases to justify running an NGS panel, and we really want an answer, we'll ask the lab scientists to do an RT-PCR, and they usually oblige us and we get a quick answer that way. Array CGH is very useful for something like neuroblastoma, and here basically this has replaced karyotyping, so we take whole DNA, you could say a mulch of DNA from the tumour. We take pooled normal DNA from a population of people who have normal DNA, and one is labeled green, one is labeled red. They're both applied to a tile which has all of the DNA arrayed on it, and the probes will stick very specifically to the DNA sequence within the genome. It's arranged chromosome by chromosome, and then you can get a readout of where you have relative excess of your tumor DNA, meaning you get, for example, if we've labeled it red as the test DNA there, you'll see red on your tiled array. If we see green, it means there's a relative deficit of the tumor, presuming that the normal green labeled is of a normal complement, and yellow is if they're equal. So this way we can work out where we have gains and losses, and this we use all the time for neuroblastoma cases to look for 1p deletion and so on, 11q gain, etc. Just a couple of caveats. So again, the top panel shows you classic Ewing sarcoma. I showed you something similar, very monotonous blue cells. The fixed tissue section, again, showing you very monotonous round cells, and a classic light cell, dark cell pattern, CD99 reactivity. Here we actually have fish that would suggest there's no break apart of just EWSR1. But if you then use EWSR1 and fly, you can show that there is a translocation. So just because you get a negative result, if you're quite sure that you are looking at what you think you're looking at, go for another technique or use a different approach with the same technique. So the break apart failed because obviously the split was somewhere else, but the come together worked because we now have the partner gene and EWSR1 at the bottom. We've got sick rearranged tumor. It looks a little different, it's not as monotonous, and we have little CD99 staining, so that again is a big clue along with WT1 staining. Again, a discrepancy here where we have EWSR1 fly1 is actually negative in this case, so that's the blue coming up extremely late. So we don't have EWSR1 fly1 picked up here, but we actually did find it by fish. So obviously the primers that we use for RT-PCR did not encompass the actual breakpoint that we wanted to demonstrate. So there are variants that occur in biology, just naturally throughout the tumor world, and one has to think of additional approaches. So I would suggest that no individual technique will cover everything, but once you understand how your assays work and know the deficiencies, if you like, or the weaknesses of the assay, and have the facility to use maybe an additional technique to confirm your thoughts, you're fine. This is what we have moved to, so this is a panel. It has both a DNA panel and an RNA panel actually, and we look for genes that are involved in solid tumours in children, and sarcomas are covered very comprehensively, so we get cases from around Ireland from adult sarcomas as well, because not that many labs have molecular. And we can obviously interrogate all of these in one go, which is very, very helpful, and then I usually go through the results. So this is John O'Brien, our chief scientist, smiling at you there. He's sitting in front of our ion torrent, and this is in the portacabin that I mentioned that we were able to get during COVID. So everything should still start with your microscope. You have to look at your H&E, you have to do the correct immunos, and then you have to think, what molecular do I want to do? Yes, of course, you can just throw your case on molecular, but I think there's a big risk that you may misinterpret things if you do it that way. So there still is a role for histology. Does it matter? Yes, it does. We need to make correct diagnoses to optimally treat our patients. That's really the role of the pathologist. It needs to be speedy, it needs to be correct. And particularly when the differential is quite tight on H&E and with immunos, you need the molecular to really resolve that. And I've shown you in the randomized sarcoma talk, you need the molecular to subcategorize more accurately as well. This includes risk stratification. There may be prognostic value to it as well. That's not really big print stuff. But increasingly, therapeutic opportunity can come from looking at your cases by molecular means as well. And we've had many discussions. Dr. Capper mentioned BRAF and MAP kinase pathway mutations and so on. So that's it. Thank you very much for your attention.
Thank you, Maureen. Once again, we're going to go march straight on to our next presentation. That's by Dr. Nguyen for outcome of osteosarcoma.